Hello, and thank you for joining me on this peaceful journey into the hidden yet fascinating world of viruses. Tonight, we'll slowly drift into a state of tranquility as we explore these tiny, elusive entities that have been part of life since its very beginning. Though invisible to the naked eye, viruses have evolved alongside every living creature, adapting with remarkable precision to the ever-changing rhythms of the world. Imagine these ancient beings, intricate in their design, quietly influencing the course of life. They don't merely invade, they shape ecosystems, influence biodiversity, and even guide the health of entire species, including our own. Viruses may appear as simple intruders, but they are much more than that. They are weavers in the tapestry of life, playing a subtle yet profound role in the balance of nature. As we delve into their evolutionary journey, consider how these microscopic travelers influence the world around us, reshaping life in ways that continue to surprise and enlighten us. From their ancient origins to their present-day impact, viruses hold secrets that guide the very course of life on Earth. If this journey resonates with you, please consider liking, subscribing, or sharing your thoughts in the comments below. Your support helps bring more curious minds into this tranquil space, and for that, I am deeply grateful. You can also turn on notifications so you never miss another moment of our explorations. But for now, simply breathe. Let your body relax, your thoughts soften, and allow your mind to float gently into this world of viruses. Let's begin. The first viruses are believed to have originated more than 3 billion years ago during the earliest stages of life on Earth. These ancient viruses likely evolved alongside the first cells, possibly from genetic material that escaped from early, primitive organisms. Despite their microscopic size, viruses play a pivotal role in shaping evolution. Over time, they've adapted and diversified, becoming more specialized in their interactions with various hosts, from bacteria to plants to humans. While some viruses cause diseases, others are beneficial, aiding in processes like gene transfer, which can drive genetic diversity in populations. Unlike other microorganisms, viruses cannot reproduce independently. They rely entirely on a host cell's machinery to replicate, making them unique among biological entities. This parasitic nature of viruses is a critical aspect of their existence. When a virus infects a host, it injects its genetic material into the cell, hijacking the cell's machinery to produce copies of itself. This process can be incredibly efficient, sometimes overwhelming the host and causing cell death, which is why viral infections can often lead to illness. There are an estimated 10 million trillion viruses on Earth, most of which remain invisible to the human eye. The majority of these viruses infect bacteria, a group known as bacteriophages. These viruses are not harmful to humans, but play a significant role in regulating bacterial populations in the environment. Without bacteriophages, bacteria could multiply unchecked, disrupting ecosystems and nutrient cycles. Interestingly, some scientists are exploring bacteriophages as alternatives to antibiotics in the fight against drug-resistant bacteria. Despite their notoriety as agents of disease, viruses are essential for the functioning of ecosystems. For example, marine viruses help regulate the balance of microbial life in the oceans. In doing so, they influence the global carbon cycle, affecting climate change. When viruses infect plankton and other marine organisms, they cause them to burst open, releasing organic material into the water, which is then consumed by other organisms in the ecosystem. This process has far-reaching implications for oceanic food chains and global carbon cycles. One of the most fascinating aspects of viruses is their ability to evolve rapidly. Because they can reproduce in vast numbers and infect many hosts, viruses are under intense evolutionary pressure. This means they can mutate quickly, leading to new strains and variants. This rapid mutation rate can make it difficult for the immune system to keep up, as seen in the ongoing battle between the human body and influenza viruses, which change every year, making a new flu vaccine necessary. Some viruses, known as retroviruses, have the unique ability to integrate their genetic material into the host's genome. HIV, the virus responsible for AIDS, is a retrovirus. By integrating itself into the host's DNA, the virus ensures its survival and can continue to replicate, even when the host's immune system tries to eliminate it. This ability to hide in the host's DNA makes retroviral infections difficult to treat and is one of the reasons why HIV remains a challenge for medical science. Viruses are not only agents of disease, they can also influence genetic evolution in positive ways. For example, viral DNA has been integrated into the genomes of many organisms, including humans. In fact, about 8% of the human genome is made up of ancient viral sequences. These viral genes, called endogenous retroviruses, may have been passed down from one generation to the next over millions of years. While some of these viral genes are harmless, others have been co-opted for important functions, such as regulating the immune system. The size of viruses varies greatly, with some being so tiny they can only be detected using electron microscopes. The smallest viruses, like the picornavirus, can be only about 20 nanometers in diameter, making them smaller than many bacteria. On the other hand, some viruses, like the pithovirus, can reach lengths of up to 1.5 micrometers. Despite their small size, viruses can have enormous effects on their hosts, making them powerful entities in the biological world. One of the most dangerous viral diseases known to humans is rabies, which is caused by a virus that affects the nervous system. The rabies virus is typically transmitted through the saliva of an infected animal, usually through bites. Once symptoms begin to appear, the disease is almost always fatal, but it can be prevented through post-exposure vaccination. The rabies virus has been known to humanity for thousands of years, and despite modern vaccines, it still claims thousands of lives annually, particularly in parts of the world with limited access to healthcare. Interestingly, some viruses are so specialized that they only infect a single species. For example, the human papillomavirus, HPV, infects only humans, and its various strains are associated with different health issues, from warts to cancer. Other viruses are even more specific, only infecting particular cell types within their host. The level of specialization shown by these viruses is a testament to the complexity of viral evolution and their ability to adapt to specific niches in the biological world. One of the most significant breakthroughs in virology occurred in 1953, when scientists James Watson and Francis Crick discovered the double helix structure of DNA. This discovery was pivotal not just for understanding genetics, but also for understanding how viruses function. The structure of DNA is crucial for replication, and many viruses, including retroviruses, hijack this mechanism to reproduce. This insight into the molecular biology of viruses laid the groundwork for the development of modern treatments and vaccines. Viruses are capable of infecting every form of life on Earth, from bacteria to humans, and they exhibit an incredible diversity of forms. There are even viruses that infect other viruses. Known as virophages, these unique entities are parasites that target larger viruses, reducing their ability to replicate. Virophages were first discovered in 2008 and are still a subject of ongoing research, further demonstrating the complexity and interconnectedness of the viral world. A viral particle is known as a virion, and it is the complete form of the virus outside of a host cell. Virions consist of a core of genetic material, either DNA or RNA, encased in a protective protein coat called a capsid. Some viruses have an additional lipid layer surrounding their capsid, known as an envelope. This envelope is derived from the host cell membrane and can help the virus evade the immune system. The structure of a virion is meticulously designed to survive in the harsh environment outside a host and to efficiently invade new cells when the opportunity arises. Incredibly, some viruses exhibit behavior that mimics living organisms. For instance, a virus known as the Mimivirus was discovered in 2003 and is notable for being one of the largest known viruses. Its size and complexity, including a genome comparable in size to that of certain bacteria, raised questions about whether viruses should be classified as living organisms. While Mimiviruses don't have the full characteristics of life, their ability to replicate and evolve challenges traditional definitions of life. Their discovery opened up a new chapter in our understanding of the boundaries between life and non-life. Not all viruses infect animals or plants. Some target bacteria, and these are known as bacteriophages. Bacteriophages, often referred to as phages, can be found in nearly every environment on Earth. They are estimated to outnumber bacteria by a factor of 10. Phages play a critical role in controlling bacterial populations, preventing overgrowth and maintaining ecological balance. Their role in nature is essential, and scientists are even exploring phages as alternatives to antibiotics, given their ability to selectively target and destroy harmful bacteria without harming beneficial microbes. The viral life cycle is divided into stages, and one of the most dramatic occurs during the lysis phase, where the infected host cell bursts open to release new viral particles. This process typically results in the destruction of the host cell, which can cause tissue damage and inflammation in the infected organism. The number of virions produced and released can vary from a few to thousands, depending on the type of virus. This explosive replication process is often what leads to the symptoms of a viral infection, such as fever and swelling, as the immune system works to combat the invasion. Some viruses have evolved sophisticated strategies to avoid detection by the immune system. For example, the herpes simplex virus, HSV, can enter a latent state after an initial infection, hiding in nerve cells for years. During this latency period, the virus is inactive and not detectable by the immune system. However, under certain conditions, such as stress or immune suppression, the virus can reactivate, leading to outbreaks. This ability to lie dormant is one reason why viral infections like HSV or chickenpox can resurface after many years, sometimes without warning. The genetic material within viruses can be incredibly diverse. Some viruses use RNA instead of DNA to encode their information, making them particularly interesting for scientists studying evolution. Retroviruses, for example, convert their RNA genome into DNA inside the host cell through an enzyme called reverse transcriptase. This DNA is then integrated into the host's genome, making retroviral infections harder to eradicate. Retroviruses are responsible for numerous diseases in humans, including HIV/AIDS, but they also provide insights into how genetic information is transferred across species and how evolution works on the molecular level. In addition to their role in disease, viruses are also used as powerful tools in biotechnology. Researchers have harnessed viruses to deliver genetic material into cells, a process known as viral vector gene therapy. This technique holds promise for treating genetic disorders, cancer, and other conditions by correcting defective genes or introducing new ones. Certain viruses, such as adenoviruses and lentiviruses, are particularly useful in this regard because of their ability to infect human cells efficiently. Though still in the early stages of development, viral vectors are already being used in clinical trials for gene therapy. Viruses also play a vital role in the environment, especially in the oceans. Marine viruses are involved in the regulation of phytoplankton populations, microscopic organisms that are critical for the global carbon cycle. These viruses infect and kill large numbers of phytoplankton, causing them to break apart and release carbon compounds into the water. The microbial cascade, triggered by viral infections in the ocean, can influence climate patterns by altering the carbon cycle and affecting the amount of carbon dioxide in the atmosphere. Viruses, in this sense, are integral to the Earth's climate system, albeit in subtle and often invisible ways. When it comes to the structure of viruses, diversity is the key. Some viruses, like the influenza virus, have an intricate shape with multiple protein spikes on their surface that allow them to bind to host cells. These spikes, known as hemagglutinins, are responsible for the virus's ability to enter cells. In fact, the constant mutations in these protein structures are why influenza vaccines must be updated each year to target the latest viral strains. The ever-changing nature of these surface proteins makes the virus highly adaptable, but also a constant challenge for public health systems. The spread of viruses often hinges on complex interactions between the virus, the environment, and its host. In some cases, viruses spread through direct contact with infected bodily fluids, while others are airborne. For instance, the measles virus is highly contagious and can linger in the air for hours after an infected person has left a room. Other viruses, such as the rhinovirus, which causes the common cold, spread through physical contact and droplets from sneezing or coughing. This wide range of transmission modes makes virus containment strategies diverse and challenging, requiring both isolation and vaccination efforts. While viral infections often cause disease, there are instances where viruses can be beneficial. For example, certain viruses are used to control populations of harmful pests, such as mosquitoes. The introduction of viruses that specifically infect mosquito species has been shown to reduce their populations in certain areas, helping to curb the spread of diseases like malaria and dengue fever. This method, known as biological control, uses viruses as a natural pest management tool, preventing the need for chemical pesticides and reducing their impact on the environment. The genome of a virus can be surprisingly simple. Some viruses consist of just a few genes, yet they can still be remarkably effective at causing infections. In contrast, some of the largest and most complex viruses, like the Mimivirus mentioned earlier, carry hundreds of genes and exhibit behaviors that resemble living organisms. This simplicity and complexity in viral genetics raise important questions about the nature of life itself. Despite their simplicity, viruses are capable of incredible diversity in their forms and effects, demonstrating the complexity of evolutionary processes at even the smallest scale. A fascinating discovery in virology was made in 2010 when scientists identified a virus that uses arsenic instead of phosphorus in its DNA. This virus, known as the arsenic bacteriophage, was found to thrive in arsenic-rich environments, challenging the long-held belief that all life requires phosphorus. This discovery expanded the understanding of the biochemical flexibility of life and hinted at the possibility of extraterrestrial life forms that could thrive under conditions very different from those found on Earth. It also underscored the adaptability of viruses, which can exploit even the most extreme environments for survival. Some viruses are capable of hijacking the immune system, turning it against the host. For example, the Epstein-Barr virus, EBV, can trigger a condition known as chronic fatigue syndrome by altering immune system responses. EBV infects B cells, a type of white blood cell, and has been linked to various diseases, including mononucleosis. Though most EBV infections are mild or asymptomatic, in some cases, the virus can remain dormant in the body and reactivate under certain conditions, leading to long-term health issues and autoimmune disorders. The human immune system is a formidable defense against viral invaders, but it is not infallible. Some viruses, like the human papillomavirus, HPV, can evade immune detection by integrating their DNA into the host's genome. This integration makes it difficult for the immune system to detect and eliminate the virus, leading to persistent infections that can result in conditions such as cervical cancer. The long-term nature of these infections is a reminder of how viruses can adapt to their hosts and find ways to bypass natural defenses, making vaccination efforts crucial in preventing some of these dangerous viral diseases. Viruses are responsible for some of the most deadly pandemics in human history. One of the most notorious is the Spanish flu of 1918, caused by an influenza virus. This pandemic infected approximately 1/3 of the world's population, leading to an estimated 50 million deaths. What made the Spanish flu particularly deadly was its ability to target healthy young adults, unlike other flu strains that primarily affect the very young or elderly. The virus's rapid mutation rate and its ability to spread quickly through air travel were key factors in its deadly spread. Not all viruses cause harm. Some can actually benefit their host. For instance, the symbiotic relationship between certain viruses and bacteria can be mutually advantageous. These bacteriophages help regulate the population of bacteria by infecting and killing them, which prevents the overgrowth of any one bacterial species that could otherwise dominate an ecosystem. In this way, viruses contribute to the balance of microbial communities in various environments, including the human microbiome, where they help control the growth of potentially harmful bacteria while leaving beneficial microbes unharmed. The complexity of viral replication is a marvel of biological engineering. Once a virus enters a host cell, it essentially hijacks the cell's machinery to produce copies of itself. However, this process is not without its challenges. Viruses must first uncoat their genetic material, ensuring it can be translated into proteins that form new viral particles. The virus then assembles these proteins and nucleic acids into new virions before releasing them to infect other cells. This highly coordinated process showcases the intricate nature of viral life, despite viruses themselves being devoid of cellular structures and metabolic functions. Despite their simplicity, some viruses are capable of extraordinary complexity in their interactions with the host. For example, the hepatitis C virus, HCV, targets liver cells and can persist in the body for years, often causing chronic infections. The virus alters the immune response, making it difficult for the body to fight off the infection. Over time, persistent HCV infections can lead to liver damage, cirrhosis, or liver cancer. The ability of HCV to remain dormant and evade immune detection illustrates the sophisticated mechanisms viruses have developed to survive within their hosts. Interestingly, some viruses are so specific to their host species that they exhibit what is known as host specificity. The Zika virus, for example, primarily infects humans and is transmitted by mosquitoes, particularly the Aedes aegypti species. Its impact on human health, particularly during pregnancy, is devastating, as it can lead to birth defects like microcephaly. The Zika virus's ability to only affect certain hosts is an example of how viruses evolve to exploit particular ecological niches, demonstrating their remarkable specialization. Many viruses are not just passive invaders. Some actively manipulate the behavior of their hosts. The rabies virus is infamous for this, as it alters the behavior of infected animals, making them more likely to bite and spread the virus. This manipulation increases the likelihood of transmission to other animals or humans. Infected animals often exhibit aggression as the virus affects their central nervous system. Such behavioral changes increase the virus's chances of spreading, showcasing the evolutionary pressure that viruses face to ensure their survival. Viral particles are remarkably durable, capable of surviving outside a host for extended periods of time. Some viruses, like the smallpox virus, can remain infectious in a dry state for years. Smallpox, once a global scourge, was eradicated through a worldwide vaccination effort, but the virus itself could persist in certain environments long after the last human infection. This resilience is a testament to the virus's ability to survive in hostile conditions, waiting for the right host to emerge and continue its life cycle. Viruses can also play a role in the horizontal transfer of genes, a process that can drive evolution. When a virus infects a host, it sometimes carries genetic material from one organism to another, introducing new genetic material into the host genome. This phenomenon, known as horizontal gene transfer, can lead to genetic diversity within populations and contribute to evolutionary changes. It is one of the ways that viruses influence not only the organisms they infect, but also the broader ecosystems in which they exist. The role of viruses in shaping biodiversity is immense, especially in the microbial world. In oceans, for example, viruses play a crucial part in the life cycle of marine organisms like plankton, where they regulate population numbers and maintain balance in the food web. Viral infections cause plankton to break apart, releasing nutrients that fuel the growth of other marine organisms. This cycle is integral to oceanic ecosystems and highlights the interconnectedness of life at a microscopic level. Without viruses, marine ecosystems might collapse due to unchecked plankton growth. Another fascinating aspect of viruses is their ability to evolve rapidly in response to environmental pressures. The influenza virus, for instance, is notorious for its ability to mutate quickly, which is why new flu vaccines need to be developed each year. The process through which this virus evolves is known as antigenic shift, where the virus's genetic material undergoes significant changes, creating new strains. This constant adaptation makes it difficult for the immune system to recognize and combat the virus, requiring humans to adapt continuously through vaccination efforts. Some viruses, like the herpes simplex virus, HSV, are capable of remaining dormant in the host for long periods, evading the immune system. These viruses can be triggered to reactivate by external stressors like illness, physical trauma, or emotional stress. This ability to lay dormant and reawaken at a later time is a survival strategy that allows the virus to persist in the host without being detected. For many people, HSV remains asymptomatic most of their lives, only flaring up during times of vulnerability, which complicates diagnosis and treatment. Viruses are not always harmful to their hosts. In some cases, they can offer protection. Certain viruses infect bacteria, protecting plants and animals from harmful bacterial infections. For instance, some phages, viruses that target bacteria, can specifically target harmful bacteria like E. coli, leaving beneficial bacteria intact. This ability to selectively target pathogens is being explored as a potential therapeutic approach to treat bacterial infections, particularly those resistant to antibiotics. Bacteriophages could offer a promising alternative to traditional antibiotics, especially in an era of increasing antibiotic resistance. Viruses are masters of survival, and they achieve this in part through their ability to evolve alongside their hosts. This coevolution can lead to the development of viral strains that are better adapted to their hosts over time. For example, the human papillomavirus, HPV, has co-evolved with humans for thousands of years, becoming highly specialized to infect the skin and mucous membranes. Its evolution has resulted in the development of different strains that vary in their ability to cause disease, with some linked to cancers, while others cause benign warts. One of the most striking features of viruses is their ability to outsmart the immune system through genetic diversity. The influenza virus, for example, can produce new viral strains that the immune system has never encountered before. This happens because the virus undergoes frequent mutations, particularly in its surface proteins, hemagglutinin and neuraminidase. These mutations allow the virus to evade immune recognition, which is why we need to get a new flu vaccine every year. The rapid mutation of influenza viruses is a reminder of how adaptable viruses are, constantly evolving to stay one step ahead of their host's defenses. Viruses can also borrow genetic material from their hosts to increase their chances of survival. This process, known as genetic recombination, allows viruses to take advantage of the host's existing genetic pool. For example, when a virus infects an organism with a compromised immune system, it may swap genetic material with the host's own cells. This recombination can lead to the creation of new viral strains that might be more effective at infecting different species or even creating new diseases. Genetic recombination is one of the reasons why some viral outbreaks can seem to appear out of nowhere. The concept of viral dormancy or latency is seen in several viruses, with the herpes simplex virus, HSV, being a prime example. After an initial infection, HSV can remain latent in nerve cells, where it stays inactive for months or even years. During this time, the virus avoids detection by the immune system. When triggered by certain factors like stress, illness, or a weakened immune system, HSV can reactivate, leading to painful outbreaks, often in the form of cold sores or genital lesions. This ability to hide and later re-emerge is a survival strategy that allows viruses to persist within their hosts for extended periods. Some viruses are known to induce what is called an immune escape mechanism. For instance, the Epstein-Barr virus, EBV, which causes mononucleosis, can alter the immune system's response to ensure its own survival. By inserting its genetic material into the host's DNA, EBV effectively tricks the immune system into ignoring the virus, allowing it to persist within the body. This immune evasion is not only a hallmark of EBV, but also other viruses like HIV, which similarly integrates its genetic material into host cells, making it harder for the body to recognize and fight off the infection. The smallpox virus, once one of the deadliest pathogens in human history, was eradicated in 1980 through a global vaccination campaign. Smallpox, caused by the variola virus, killed an estimated 300 million people in the 20th century alone. The virus was highly contagious, spreading through respiratory droplets, and caused fever, skin lesions, and often death. The global eradication of smallpox is considered one of the greatest achievements in public health history, demonstrating the power of vaccines in controlling infectious diseases. Today, smallpox remains the only human disease that has been completely eradicated from the planet. Viruses can have a profound impact on ecosystems, particularly in aquatic environments. In oceans, viruses target phytoplankton, which are the foundation of marine food webs. By infecting and killing these tiny organisms, viruses help regulate their populations, preventing any one species from dominating the ecosystem. This process not only helps maintain biodiversity, but also has implications for the global carbon cycle. When viruses cause plankton to burst open, they release organic material that is consumed by other organisms, influencing nutrient cycles and playing a significant role in oceanic food chains. One of the ways that viruses manipulate their hosts is by altering the way cells communicate. Viruses can hijack cellular signaling pathways, reprogramming the host cell to favor viral replication. For example, the human papillomavirus, HPV, alters the cell cycle by inactivating tumor suppressor proteins like p53. This manipulation can lead to uncontrolled cell division, which in turn may result in the formation of tumors, including cervical cancer. The ability of viruses to influence cellular processes is a testament to their complex nature and their deep integration into the biology of life on Earth. While most viruses are harmful to their hosts, some can actually confer benefits. For example, certain bacteriophages can help protect plants from harmful bacterial infections. These viruses specifically target bacterial pathogens, helping to prevent diseases like bacterial blight in crops. In this way, bacteriophages act as natural pest control agents, reducing the need for chemical pesticides. This method of biological control is becoming increasingly important in sustainable agriculture, as scientists explore ways to use viruses to protect crops and reduce the environmental impact of farming. The way viruses replicate can be influenced by their environment, making some viruses more potent in certain conditions. Temperature, humidity, and even the host's immune status can all impact viral replication. For example, the rhinovirus, which causes the common cold, replicates more efficiently at cooler temperatures, which is why colds are more common in winter months when people are indoors and the air is colder. The adaptation of viruses to their environment underscores the dynamic relationship between virus and host, and how environmental factors can shape the course of infection. Some viruses can have surprising uses in medical research and therapy. Gene therapy, for example, often relies on modified viruses to deliver new or corrected genetic material into a patient's cells. Viruses such as adenoviruses and lentiviruses are modified to carry therapeutic genes, which they deliver into human cells during infection. These viruses are stripped of their ability to cause disease, making them effective and relatively safe tools for treating genetic disorders like cystic fibrosis and muscular dystrophy. Viral vectors are opening up new avenues for treating diseases that were once considered untreatable. A particularly intriguing aspect of viruses is their role in shaping the microbial communities of the human body. The human microbiome, which consists of trillions of bacteria, fungi, and other microbes, is influenced by viruses. Phages that target specific bacteria within the microbiome can help maintain a healthy balance of microbes. Disruptions in this delicate balance, often caused by antibiotics or viral infections, can lead to conditions like dysbiosis, which is linked to diseases such as irritable bowel syndrome and autoimmune disorders. Viruses, by influencing the composition of the microbiome, indirectly affect human health in profound ways. The process of viral infection begins when a virus attaches to a host cell using specific proteins on its surface. This attachment is highly specific, with some viruses only able to infect particular species or even specific tissues within an organism. For example, the HIV virus only infects immune cells with a particular receptor, while the polio virus targets nerve cells. This specificity allows viruses to be incredibly efficient at infecting their chosen host cells, but it also means that viruses can be highly specialized and difficult to study as they rely on intricate interactions with their environment. The transmission of viruses can be incredibly varied. While many viruses, like the influenza virus, are airborne, others rely on vector organisms, such as mosquitoes, to spread. The Zika virus, for instance, is primarily transmitted through the bite of an infected Aedes mosquito. This mode of transmission has made it difficult to control outbreaks, as it requires addressing not only the viral infection, but also the mosquito population. Understanding the vectors of viral transmission is crucial in developing effective prevention strategies, especially in regions where vector-borne diseases are endemic. Some viruses have evolved mechanisms that allow them to jump between species. This phenomenon, known as zoonotic transmission, occurs when a virus is able to infect a species it hasn't previously affected. One well-known example of this is the emergence of the SARS-CoV-2 virus, which caused the COVID-19 pandemic. Initially believed to have originated in bats, the virus likely jumped to humans through an intermediate animal host. Zoonotic diseases like this have had significant impacts on human history, as viruses adapt to new species and begin to spread across populations, often resulting in widespread disease outbreaks. The size of a virus can vary dramatically depending on the type. The largest known virus is the Mimivirus, which measures over 700 nanometers in diameter. This is enormous compared to the smallest viruses, such as the picornaviruses, which can be as small as 20 nanometers. Despite their small size, viruses can cause significant disease and play a powerful role in nature. The size variation between different viruses highlights the remarkable diversity in viral structures and how each is specifically adapted to infect its chosen host. One of the most unusual viruses is the Pandora virus, a virus so large that it blurs the lines between living organisms and viruses. The Pandora virus has a genome with over 2,500 genes, which is far larger than many bacteria. It was discovered in 2013 in a lake in Chile. This virus challenges our understanding of what constitutes a true virus and raises intriguing questions about the evolutionary relationships between viruses and other forms of life. Pandora virus and other giant viruses are altering the way scientists think about the boundary between living organisms and viruses. Another fascinating aspect of viruses is their ability to decide whether to undergo the lytic cycle or the lysogenic cycle. The lytic cycle is the one where the virus hijacks the host cell to replicate and then causes the cell to burst, releasing new virions. On the other hand, the lysogenic cycle allows the viral genome to be incorporated into the host cell's DNA, becoming a dormant part of the host's genome. The decision between these two paths is influenced by environmental factors and the state of the host cell, adding an element of unpredictability to the behavior of viral infections. Some viruses are so specific that they can only infect certain types of cells. For example, the polio virus targets nerve cells, causing the disease polio. Similarly, the measles virus targets immune cells, making it highly contagious and sometimes deadly. The specificity of viral infections highlights the evolutionary strategies viruses use to ensure their survival. By targeting specific cell types, viruses can exploit the host's biological processes to reproduce efficiently while evading immune responses. This precision is one of the reasons why viral infections can have such a wide range of effects, from mild to fatal. Viruses can also influence the evolution of the organisms they infect. A process called viral evolution occurs as viruses constantly adapt their hosts in response to environmental changes. This evolutionary arms race can lead to new viral strains that are better able to evade the immune system. For instance, the HIV virus has evolved rapidly over time, developing resistance to antiviral drugs and immune defenses. This ability to rapidly adapt makes viral diseases like HIV/AIDS difficult to treat and demonstrates the complex relationship between viruses and the immune systems of their hosts. A particularly interesting feature of certain viruses is their ability to modify the genetic makeup of their hosts. Some viruses, like retroviruses, insert their genetic material directly into the host's DNA. HIV is one example of a retrovirus that uses reverse transcriptase to convert its RNA into DNA, which then becomes integrated into the host cell's genome. This integration allows the virus to persist in the body for years, leading to chronic infections. The ability of viruses to alter their host's genetic material not only ensures their survival, but also contributes to the complexity of the interactions between viruses and their hosts. Viruses play a major role in the global nutrient cycle, particularly in the oceans. Marine viruses infect and kill vast numbers of marine microorganisms, including plankton. This process is important because it releases organic material into the water that other organisms can consume. This cycling of nutrients helps to maintain balance in marine ecosystems and even affects the global carbon cycle. Without viruses, plankton populations could become too large, disrupting the delicate balance of ocean life and potentially impacting the Earth's climate. Despite their small size, viruses are capable of causing massive disruptions in ecosystems. For example, the chytrid fungus, which is technically a type of virus, has devastated amphibian populations worldwide. This virus infects the skin of amphibians, disrupting their ability to absorb water and regulate their bodily functions. The chytrid fungus has been responsible for the extinction or near extinction of numerous species, showing how viruses can have far-reaching consequences for biodiversity. Such outbreaks highlight the fragile nature of ecosystems and the powerful role viruses play in shaping the natural world. The hepatitis B virus, HBV, is a particularly well-studied example of a virus that can cause chronic liver disease, including cirrhosis and liver cancer. Despite the availability of a vaccine to prevent HBV, millions of people around the world remain chronically infected. Hepatitis B is transmitted through contact with infected blood or bodily fluids, making it highly contagious. The virus can persist in the liver for years, often without symptoms, until it causes serious liver damage. Understanding the mechanisms by which HBV causes liver damage has been crucial in developing antiviral treatments and vaccines. One remarkable aspect of certain viruses is their ability to escape immune detection through a process known as antigenic variation. The influenza virus, for example, changes the structure of its surface proteins in a way that allows it to evade the immune system. This variation is why people can get the flu more than once, and why the flu vaccine needs to be updated annually. The virus effectively hides from the immune system by altering its appearance, making it difficult for the body to recognize and fight the infection. Viruses are capable of rapidly adapting to new environments. When viruses mutate, they can acquire new properties, such as resistance to antiviral drugs or the ability to infect new species. The emergence of drug-resistant strains of viruses like HIV and the influenza virus has posed a significant challenge for medical professionals. These adaptive capabilities are what make viruses so dangerous and difficult to control. It's also why antiviral treatments and vaccines must constantly evolve to keep pace with new viral strains. The discovery of viruses is credited to the Russian scientist Dmitry Ivanovsky, who in 1892 was investigating a plant disease in tobacco. Ivanovsky discovered that the agent causing the disease could pass through a filter that was fine enough to trap bacteria. This observation suggested that the pathogen responsible was not a bacterium, but something far smaller, leading to the eventual identification of viruses. This groundbreaking discovery opened up a new field of study in microbiology and helped lay the foundation for our understanding of viruses as entities distinct from bacteria. The ability of viruses to evolve so quickly is a result of their unique replication process. Unlike most organisms, viruses do not replicate by dividing cells. Instead, they hijack the machinery of a host cell to create new viral particles. Because viruses have such high mutation rates, especially RNA viruses like the flu or HIV, they can adapt quickly to new environments or host defenses. This rapid evolution is one of the reasons viruses can be so difficult to eradicate, as new variants constantly emerge, rendering previous treatments or vaccines less effective. Some viruses are so small that they can be difficult to detect with traditional laboratory equipment. The smallest known viruses, such as the picornavirus family, are about 20 to 30 nanometers in diameter. These microscopic agents are capable of slipping past many of the body's defenses and can go undetected for extended periods. Despite their small size, these viruses can cause a range of diseases, from the common cold to polio, demonstrating that size does not limit the potential impact of viruses on human health. There is a curious phenomenon in the study of viruses known as virus-like particles, VLPs. These are particles that resemble viruses but do not contain any viral genetic material. They are often used in vaccine development, where they stimulate an immune response without causing illness. For instance, the HPV vaccine uses VLPs to protect against the human papillomavirus. The use of virus-like particles has provided an innovative way to develop vaccines, without the risks associated with using live viruses, revolutionizing vaccine design and leading to the successful prevention of various diseases. Some viruses are capable of altering the genetics of their host cells in dramatic ways. Retroviruses, such as HIV, use an enzyme called reverse transcriptase to convert their RNA into DNA. Once the viral DNA is integrated into the host cell's genome, it can remain dormant for years, creating a permanent reservoir of the virus within the host. This ability to alter the host genome is one of the reasons why retroviruses can be so challenging to treat. By the time the virus begins to replicate again, it may have already spread to other cells, complicating efforts to combat it. The study of viruses has led to major medical advancements, including the development of targeted therapies. For example, scientists are exploring how to use viruses to deliver targeted treatments for cancer. These so-called oncolytic viruses selectively infect and kill cancer cells while leaving healthy cells unharmed. Oncolytic virotherapy is still in its experimental stages, but early research shows promising results, particularly with viruses like the herpes simplex virus, HSV. If proven effective, this treatment could become a powerful weapon in the fight against cancer. Viruses can be used to manipulate genetic material in ways that would be impossible using traditional genetic modification methods. One example is the use of adeno-associated viruses, AAV, as vectors for gene therapy. These viruses are engineered to carry therapeutic genes into human cells, offering a potential cure for genetic disorders like cystic fibrosis and muscular dystrophy. AAVs are highly specific, targeting only certain types of cells, which makes them an ideal tool for gene delivery. This has opened up new possibilities for treating diseases that have long been considered incurable. The shape of a virus is often determined by the arrangement of its protein subunits. Many viruses, like the icosahedral viruses, have a symmetrical shape, with proteins arranged in a regular geometric pattern. This geometric symmetry is not just an aesthetic feature. It is a highly efficient design that allows the virus to package its genetic material into a compact, sturdy capsule. The icosahedral shape provides stability to the virus and ensures that it can survive the often harsh environments outside of a host cell, waiting for an opportunity to infect. In the world of viruses, the concept of quasi-species is important for understanding how viral populations evolve. Quasi-species refer to the diverse collection of virus variants within a single host. Even a single viral infection can consist of many different versions of the same virus, each with slight genetic variations. This genetic diversity allows viruses to rapidly adapt to changes in their environment, such as the immune system's response or antiviral drugs. Quasi-species are a key reason why viruses like HIV and influenza can evolve resistance to treatments so quickly. Viruses can also be crucial in the study of human genetics. By introducing viral genetic material into human cells, scientists are able to observe how genes interact with one another and how certain mutations might cause diseases. This practice, known as genetic transduction, has allowed for groundbreaking discoveries in medicine, including insights into cancer, neurodegenerative diseases, and even aging. Viruses are often the vehicles for studying genetic material in a controlled way, helping researchers identify gene functions and potential therapeutic targets. Bacteriophages, viruses that infect bacteria, are being explored as a solution to the growing problem of antibiotic resistance. Bacteriophages are highly specific in which bacteria they infect, meaning they can target harmful bacteria without affecting beneficial microbes. In recent years, scientists have begun to explore phage therapy as an alternative to antibiotics, especially for infections caused by antibiotic-resistant bacteria. Although still in its infancy, this approach could revolutionize medicine and provide a new weapon against multi-drug-resistant infections. Despite their microscopic size, viruses can have far-reaching effects on ecosystems. For example, in the oceans, viral infections can regulate plankton populations. When viruses infect and kill plankton, they release organic matter that other marine organisms can consume, which plays a vital role in the marine food chain. These viral interactions in marine ecosystems also help regulate the carbon cycle, contributing to global climate processes. Viruses, though often invisible to the naked eye, are key players in ecological balance and carbon sequestration. The smallest known virus, circovirus, is a tiny agent that can measure just 17-20 nanometers in diameter. Despite its small size, circovirus can infect various animals, including birds and pigs, causing diseases that impact livestock industries. While it may seem insignificant compared to larger viruses, circovirus has an outsized impact on agriculture. This highlights an interesting aspect of viruses. Size is not an indicator of their potential impact. Some of the smallest viruses in nature can cause devastating diseases, particularly in animals, plants, and even humans. Viruses can also serve as natural regulators of microbial populations. In soil ecosystems, for example, viruses help control bacterial populations, preventing any one species from overwhelming the ecosystem. This process helps maintain biodiversity in microbial communities, which in turn affects plant health and overall soil quality. By controlling bacterial populations, viruses play a critical role in ensuring that no one species dominates, thus maintaining the delicate balance that supports life in soil ecosystems. Viruses are capable of altering the natural balance of the immune system in fascinating ways. The human immunodeficiency virus, HIV, specifically targets T helper cells, which are vital to orchestrating immune responses. By attacking these crucial immune cells, HIV weakens the immune system, leading to acquired immunodeficiency syndrome, AIDS. The virus's ability to integrate its RNA into the host's DNA and persist in a latent state is why the infection remains chronic. This tactic of stealthily altering immune function allows the virus to evade complete eradication, complicating efforts to cure the disease and making the development of vaccines challenging. The interaction between viruses and their hosts can sometimes lead to unexpected outcomes, such as genetic innovation. For example, viruses can introduce new genetic material into the genomes of their hosts through a process called horizontal gene transfer. This process has been particularly evident in bacteria, where bacteriophages can transfer genes for antibiotic resistance between different bacterial strains. Such viral-induced gene transfer has profound implications for human health, particularly in the context of antibiotic resistance. Viruses are inadvertently shaping microbial evolution and, in turn, the bacterial threats we face. A unique class of viruses known as satellite viruses cannot replicate independently. Instead, they require the presence of a helper virus to replicate and propagate. These viruses are often considered parasites of other viruses, relying on them for replication machinery. An example of this is the satellite tobacco necrosis virus, which requires the tobacco mosaic virus for reproduction. The relationship between satellite and helper viruses illustrates the incredible diversity and complexity in the viral world, where different species can form intricate networks of dependency for survival. In the case of the bacteriophage, viruses can be used not only to study genetics, but also to treat bacterial infections. Bacteriophages are highly specific, targeting individual bacterial species without affecting others, making them a powerful tool in combating bacterial diseases. This specificity allows phage therapy to target antibiotic-resistant bacteria, providing an alternative treatment method in the face of growing resistance to conventional antibiotics. The resurgence of interest in phage therapy is an exciting development, especially in light of the global health crisis caused by multi-drug-resistant bacteria. Viruses often serve as vehicles for the spread of infectious diseases across species, contributing to the emergence of new illnesses. The Ebola virus, for example, is thought to have originated in bats before jumping to humans, likely through the consumption or handling of infected animals. This kind of interspecies transmission, known as zoonotic spillover, is responsible for many viral outbreaks in human populations. As deforestation and urbanization increase, the likelihood of such zoonotic transmissions occurring also rises, creating greater risks for the spread of novel viruses. Viruses exhibit remarkable adaptability, allowing them to thrive in diverse environments. Some viruses, such as the T4 bacteriophage, have evolved to exist in extreme conditions, surviving in high temperatures, acidic environments, and even in the deep oceans. These extremophilic viruses are not only fascinating because of their resilience, but also because they help scientists understand the limits of life. Their ability to survive under extreme conditions is opening new avenues for biotechnology, as researchers explore how these viruses could contribute to the development of new materials, medicines, and technologies. The concept of virus evolution also includes the viral ability to exchange genetic material with other viruses. This exchange can occur through recombination or assortment, processes in which viral genetic material from different strains is mixed. This leads to the creation of novel viral variants that may have enhanced abilities to infect hosts or evade immune defenses. An example of this is the seasonal flu, where new strains arise each year as the influenza virus recombines with other viruses, which is why flu vaccines must be updated regularly to accommodate these changing strains. One of the most notorious viruses in the history of human pandemics is the H1N1 influenza virus, which caused the Spanish flu of 1918. This virus spread rapidly across the globe, infecting nearly a third of the world's population and killing millions. What made the Spanish flu so deadly was its ability to infect healthy young adults, rather than just the very young or elderly, as is typical for most influenza strains. The virus's high mutation rate and rapid spread, aided by the movements of soldiers during World War I, made it a devastating force in global health. Interestingly, viruses can also contribute to the discovery of new biological phenomena. For example, the hepatitis C virus, HCV, once a poorly understood pathogen, has been instrumental in advancing our knowledge of viral replication and cellular biology. HCV's complex interaction with the host liver cells has provided valuable insights into how viruses hijack cellular processes to replicate. The study of HCV has also led to breakthroughs in antiviral treatments, most notably the development of direct-acting antivirals, DAAs, which have revolutionized the treatment of chronic hepatitis C infections. In an extraordinary example of viral resilience, the herpes simplex virus, HSV, has adapted over millennia to coexist with its human host. After an initial infection, HSV becomes latent, hiding in nerve cells where it remains dormant until it is reactivated by triggers such as stress or illness. During its dormant phase, the virus is almost undetectable by the immune system, allowing it to persist in the body for years or even a lifetime. This ability to remain hidden and later reactivate makes HSV a lifelong presence for many individuals, leading to recurrent outbreaks, typically in the form of cold sores. Another fascinating aspect of viruses is their potential to help us solve global problems. For instance, viruses are being explored as potential agents for cleaning up environmental pollutants. Bacteriophages, which target specific bacteria, could be used to break down harmful waste products or even degrade toxins in contaminated environments. This bioremediation strategy is still in the experimental stages, but holds promise for addressing some of the planet's most pressing environmental challenges, such as oil spills or toxic chemical contamination. Viruses have a unique ability to remain infectious for extended periods, sometimes outside of a host. The smallpox virus, for instance, was capable of surviving on surfaces for days, even in dry conditions. This survival mechanism is what made the virus so difficult to eradicate, as it could remain dormant in the environment, waiting for the opportunity to infect a new host. Understanding how viruses can endure for so long in the absence of a host has important implications for controlling the spread of infectious diseases, particularly in the context of public health. Some viruses, particularly those that infect plants, can cause significant agricultural damage. The tomato spotted wilt virus, TSWV, for example, is a major pest in agricultural regions, affecting crops such as tomatoes, peppers, and peanuts. The virus is spread by thrips, tiny insects that carry the virus from plant to plant. Once infected, plants can exhibit symptoms such as wilting, spotting, and stunted growth, leading to crop loss. The spread of plant viruses has become an increasing challenge for food security, highlighting the interconnectedness of viruses and agricultural ecosystems. One of the most remarkable features of viruses is their incredible diversity. A single ecosystem, such as a patch of ocean or a human body, can host thousands of different viruses, each infecting specific species of bacteria, fungi, plants, or animals. For instance, the human gut alone is home to trillions of bacteriophages, which are viruses that infect bacteria. These viruses play a crucial role in shaping the microbial ecosystem of the gut, influencing digestion, immunity, and even mood. This invisible viral world demonstrates the interconnectedness of life and the role viruses play in maintaining ecological balance. Some viruses are so large that they have been classified as giant viruses. These enormous viral particles challenge our conventional understanding of viruses as tiny, simple entities. The Mimivirus and Pandora virus, for example, are larger than some bacteria and have genomes that contain thousands of genes. Their size and complexity raise interesting questions about the origins of viruses and whether they could be considered a form of life in their own right. The discovery of giant viruses has blurred the lines between viruses, bacteria, and even cellular life, prompting a re-evaluation of what constitutes a living organism. The hepatitis B virus, HBV, which causes chronic liver disease, is unique in that it has a circular genome. This circular structure is part of what makes the virus so effective at replicating within its host. The ability of HBV to persist in the liver for years without triggering a strong immune response allows it to remain dormant until reactivation. This long-lasting infection can eventually lead to cirrhosis or liver cancer, making HBV one of the most significant viral pathogens in terms of global health. Vaccines against HBV have drastically reduced its impact, but millions of people still live with chronic infection. Some viruses, like the influenza virus, are highly contagious and can spread quickly through human populations, especially during the colder months. Influenza viruses spread primarily through respiratory droplets when an infected person coughs or sneezes. They can also survive on surfaces for several hours, making it easy for the virus to spread in crowded spaces like schools, offices, and public transportation. The seasonal nature of flu outbreaks is thought to be linked to environmental factors, such as lower humidity, which allows the virus to thrive. Despite the availability of vaccines, the flu continues to cause widespread illness each year. Viruses can cause chronic infections that persist for a lifetime. Hepatitis C virus, HCV, for example, can remain in the liver for decades, silently causing damage before any symptoms appear. Chronic HCV infection is a leading cause of cirrhosis and liver cancer. Unlike other viruses that cause acute infections with a clear resolution, HCV can evade the immune system for years, making it difficult to treat. Fortunately, recent advancements in antiviral treatments have revolutionized the way HCV is managed, offering the possibility of a cure for many individuals infected with the virus. The study of bacteriophages, viruses that infect bacteria, has revealed some fascinating facts about the relationship between viruses and their bacterial hosts. Bacteriophages are often so specific that they only infect particular strains of bacteria, leaving beneficial bacteria unharmed. This specificity has made bacteriophages valuable tools in medical and environmental applications. For example, phages are being explored as alternatives to antibiotics in treating antibiotic-resistant infections. Additionally, phages are used in agricultural settings to control bacterial diseases in crops, showing that viruses can be harnessed for beneficial purposes. In addition to causing disease, viruses are being used in innovative ways to advance scientific research. One example is the use of adenoviruses in gene therapy. By modifying these viruses to carry therapeutic genes, researchers can deliver genetic material directly into cells. This method is being explored as a potential treatment for genetic disorders like cystic fibrosis and muscular dystrophy. The ability of viruses to insert their genetic material into host cells makes them an ideal tool for genetic modification, paving the way for treatments that could change the course of genetic diseases. The influenza virus has an astonishing ability to change its genetic makeup over time, making it one of the most unpredictable and challenging viruses to control. This phenomenon, known as antigenic drift, occurs when small genetic changes accumulate in the virus's surface proteins, making it difficult for the immune system to recognize and fight the infection. In addition, the influenza virus can undergo antigenic shift, a more dramatic genetic change that occurs when two different strains of the virus combine to form a new strain. This process is responsible for the emergence of novel flu strains that can lead to pandemics. Viruses are not always harmful to their hosts. In fact, some viruses can have a beneficial effect on the organisms they infect. One of the most well-known examples is the relationship between certain viruses and coral reefs. Some viruses that infect marine bacteria play a crucial role in maintaining the health of coral reefs by regulating bacterial populations. Without these viruses, harmful bacteria could overpopulate, potentially leading to coral disease. This example highlights the complex and sometimes symbiotic relationships that viruses can have with their hosts, contributing to ecological balance. One of the most unique aspects of viral behavior is their ability to manipulate the host cell's machinery to their advantage. When a virus infects a cell, it hijacks the cell's replication machinery to produce new viral particles. In the case of retroviruses, such as HIV, the virus integrates its RNA into the host cell's DNA, ensuring that the virus's genetic material is passed on to new cells when they divide. This integration allows the virus to persist in the host for extended periods, often causing chronic infections. The ability of viruses to manipulate host genetics is a key feature that contributes to their survival and persistence. Despite being microscopic, viruses can have a massive impact on the health of entire populations. Measles, for instance, is a highly contagious viral disease that spreads rapidly through respiratory droplets. Before vaccines were widely available, measles outbreaks were responsible for millions of deaths worldwide. Even today, in regions where vaccination rates are low, measles continues to be a significant public health threat. The highly contagious nature of the virus is one reason why vaccination campaigns are so important, as herd immunity can help prevent the spread of diseases like measles in vulnerable populations. Viruses can sometimes be used to study fundamental biological processes. The study of bacteriophages has contributed significantly to our understanding of molecular genetics and DNA. Bacteriophages were among the first viruses to be studied in detail, and they played a pivotal role in the discovery of DNA as the molecule of heredity. By observing how phages infect bacterial cells and replicate their genetic material, scientists were able to develop techniques that would later be used in gene cloning and recombinant DNA technology, which are foundational tools in modern molecular biology. Some viruses can actually change the physical properties of their hosts in dramatic ways. For instance, the rabies virus alters the behavior of infected animals, often causing them to become aggressive or unusually friendly. This alteration in behavior increases the likelihood that the infected animal will bite and transmit the virus to others. In this way, the virus is able to spread more efficiently, demonstrating how viruses can influence not just the biology, but also the behavior of their hosts, optimizing their chances of transmission. Viruses can have a profound effect on the ecosystems they invade. In the case of bacteriophages, viruses that infect bacteria, they are integral to the microbial food web, controlling bacterial populations in environments like soil and oceans. These viruses infect and kill bacteria, releasing nutrients that are then available for other microorganisms. This process helps maintain a balance in microbial communities, preventing any one species from overgrowing. In ocean ecosystems, bacteriophages help regulate bacterial blooms, which, if left unchecked, could disrupt the balance of the entire marine ecosystem. These viruses, while invisible to the naked eye, play a key role in global nutrient cycling. Interestingly, some viruses are able to switch between different hosts. The avian influenza virus, for example, can infect both birds and humans, making it a potential threat for cross-species transmission. While this virus primarily affects birds, it has the potential to jump to humans under the right conditions, as seen in the 2009 H1N1 pandemic. The ability of viruses to infect multiple species is one of the reasons they pose such a danger, as they can quickly evolve to infect new hosts and spread more widely, often with devastating consequences for public health. A fascinating aspect of viral replication is the molecular machinery viruses employ to hijack host cells. After a virus enters a host cell, it essentially takes over the cell's metabolic processes. For instance, the Zika virus, known for its role in causing birth defects in humans, uses the host's endoplasmic reticulum to assemble new viral particles. The virus manipulates the host's cellular structures to create the perfect environment for its replication. This ability to hijack cellular machinery is why viruses are often considered obligate intracellular parasites. They rely entirely on the host cell to reproduce. The concept of viral latency is a remarkable survival strategy used by certain viruses, including the herpes simplex virus, HSV. After an initial infection, HSV can lie dormant in nerve cells for years, only to reactivate later under specific conditions, such as stress or illness. This ability to hide from the immune system during the latent phase is what makes certain viral infections so difficult to treat. The reactivation of latent viruses can lead to recurrent outbreaks, often without warning, making it challenging for individuals with these viruses to manage their condition over the long term. In an unusual twist, some viruses may help their hosts defend against other, more harmful pathogens. For example, certain bacteriophages, which target specific bacteria, can provide a defense against harmful bacterial infections. This is especially significant in the context of antibiotic resistance, where traditional antibiotics are becoming less effective. Bacteriophage therapy, the use of viruses to fight bacterial infections, is a promising alternative treatment. Phages are highly specific, meaning they target only harmful bacteria while leaving beneficial microbes unharmed, which could help restore the balance of the microbiome without causing adverse side effects. The discovery of phage therapy, using viruses to treat bacterial infections, has renewed interest in these microscopic agents as therapeutic tools. The first successful use of bacteriophages in medicine occurred in the early 20th century, when doctors treated a variety of bacterial infections with phages. However, the rise of antibiotics pushed phage therapy into obscurity. Now, As antibiotic resistance has become a major global health issue, bacteriophages are making a comeback. Researchers are testing phage cocktails, which combine several different types of phages to target a broad range of bacteria as potential treatments for hard-to-treat infections. Despite their microscopic size, viruses have a massive influence on the planet's biodiversity. In fact, viruses are a driving force in the evolution of other organisms. As they infect bacteria, viruses introduce new genetic material into the microbial gene pool through a process known as horizontal gene transfer. This exchange of genetic information can lead to the evolution of new bacterial strains, some of which may have enhanced survival mechanisms. This ongoing process of viral infection and genetic exchange has helped shape the microbial world, influencing everything from bacterial resistance to new genes and traits in various ecosystems. Viruses can often be incredibly difficult to diagnose, especially when they cause symptoms similar to other diseases. For instance, the symptoms of a viral infection like the common cold are often indistinguishable from those caused by bacterial infections or allergies. This presents a challenge for doctors who must carefully distinguish between different causes of illness in order to recommend the correct treatment. Rapid diagnostic tests are constantly being developed to help identify viral infections faster and more accurately. But the constantly evolving nature of viruses continues to make them difficult to track and treat. The immune system's response to viral infections is an intricate and complex process. Upon encountering a virus, the body's immune system launches a series of defensive measures, starting with the recognition of viral proteins by immune cells. These immune cells then trigger the production of antibodies, which bind to the virus and neutralize it. In some cases, viruses can evade these immune responses by mutating their proteins or entering a latent state, as seen in the case of HIV. The ability of viruses to evolve and adapt to the immune system is one of the reasons why they remain such a persistent threat. Some viruses can even alter the behavior of their host organisms to facilitate their spread. For example, the rabies virus is known to cause changes in the behavior of infected animals. Rabies often leads to increased aggression, which increases the likelihood of transmission through bites. The virus has evolved to influence its host's behavior in such a way that promotes the spread of infection. This manipulation of host behavior is a sophisticated survival strategy, ensuring that the virus can spread efficiently through a population, even if it results in the death of the infected host. Viruses can cause diseases with varying degrees of severity, from mild to fatal. The chickenpox virus, or varicella zoster for instance, causes a typically mild illness in children, characterized by itchy skin rashes and fever. However, the virus can have serious consequences for adults, especially pregnant women and immunocompromised individuals, potentially leading to complications like pneumonia or encephalitis. Even after recovery, the virus remains dormant in nerve cells and can reactivate later in life, causing shingles, a painful condition. This dual nature, causing both acute and chronic disease, makes chickenpox a fascinating subject for study. One of the most well-known examples of a viral mutation leading to significant public health consequences is the emergence of the Zika virus. This virus, which is transmitted primarily by Aedes mosquitoes, was linked to a dramatic increase in birth defects, including microcephaly in babies born to infected mothers. The rapid spread of Zika, especially across the Americas, highlighted the challenges posed by viruses in the context of global travel and climate change. As mosquito populations shift and expand, diseases like Zika, dengue, and malaria are becoming an increasing concern in areas that were previously unaffected. Viruses are masters of efficiency when it comes to replication. In just a few hours, a single virus particle can create hundreds or even thousands of new viral copies. This rapid production is made possible by the virus's ability to hijack the host cell's machinery, forcing it to prioritize viral reproduction over the cell's normal functions. The viral replication process varies depending on the type of virus. For example, DNA viruses like the herpes simplex virus replicate their genomes in the host's nucleus, while RNA viruses like the flu take over the cell's cytoplasm. This variation in replication strategies further adds to the complexity of viral behavior. While viruses themselves do not possess the biological machinery for metabolism or energy production, they are often described as molecular parasites. To reproduce, they must rely on the host's metabolic processes. This parasitic relationship is central to how viruses spread. By co-opting host resources for their own replication, viruses leave little trace behind, making them incredibly efficient and difficult to target. The evolutionary strategy of viruses as obligate intracellular parasites ensures that they thrive without needing to perform the energy-intensive functions of living organisms themselves. Some viruses can exhibit a strange and intricate relationship with their hosts that borders on mutualism, benefiting both the virus and the host in ways that seem counterintuitive. An example of this can be seen in the lysogenic cycle of certain bacteriophages. In this phase, the virus integrates its genetic material into the host bacterium's genome, where it can stay dormant for extended periods. While this doesn't immediately harm the host, the bacterium gains the ability to express certain advantageous traits provided by the virus. This kind of relationship shows the subtle ways in which viruses can influence and coexist with their hosts over long periods. One of the most visually stunning viruses is the cowpox virus, which is similar to the smallpox virus. Cowpox causes blister-like lesions on the skin of infected animals, but it has been historically significant due to its role in the development of the first smallpox vaccine by Edward Jenner in the 18th century. Jenner discovered that exposure to the less virulent cowpox virus provided immunity to the deadly smallpox virus, marking a pivotal moment in medical history. The discovery not only saved countless lives, but also laid the foundation for the field of immunology and modern vaccines. The ability of viruses to cross species barriers and infect new hosts is one of their most alarming traits. The SARS-CoV-2 virus, responsible for the COVID-19 pandemic, is a prime example. It is believed to have originated in bats, then jumped to humans via an intermediary animal host. The virus's high mutability and ability to infect multiple species make it a constant threat to public health. Cross-species transmission remains one of the most concerning factors in viral evolution, as it can lead to pandemics, particularly when a new virus finds its way into a human population without immunity. Interestingly, viruses can also play an essential role in shaping the diversity of life on Earth. In ecosystems ranging from the deep sea to the human body, viruses help regulate populations by infecting and killing bacteria. This process influences the genetic diversity of microbial communities and, by extension, the diversity of organisms that rely on them. In the oceans, for example, viruses kill billions of bacteria every day, releasing organic matter that other marine life can consume. These viral processes are critical in maintaining ecological balance, even though they are largely invisible to the human eye. Viruses often employ intricate methods to evade the immune system of their host. Influenza viruses, for instance, can change their surface proteins through a process known as antigenic variation, which prevents the immune system from recognizing and neutralizing the virus. This ability to undergo constant change is why the flu virus is so challenging to control, as new strains emerge regularly. Similarly, HIV evades immune detection by constantly mutating its surface proteins and by integrating into the host's DNA, making it difficult for the immune system to mount an effective defense. This viral evasion highlights the constant arms race between viral evolution and the immune system. The use of viruses in medicine is becoming more common, particularly in the field of gene therapy. One of the most widely used viruses for this purpose is the adeno-associated virus, AAV, which is particularly useful for delivering therapeutic genes to specific cells. AAVs are preferred because they don't cause disease in humans and can effectively target a variety of tissues. The virus is engineered to carry the desired gene into cells, where it can be incorporated into the host genome. This approach has shown promise in treating genetic disorders such as hemophilia, muscular dystrophy, and certain types of blindness. In addition to their role in disease, viruses can sometimes become dormant in their host for extended periods. Herpes simplex virus, HSV, for example, can remain dormant in nerve cells for years, only reactivating when certain triggers occur, such as stress or illness. When dormant, the virus is essentially invisible to the immune system, avoiding detection and maintaining a stealthy presence in the host. The ability of viruses like HSV to establish latency is one of the reasons why conditions caused by these viruses are so persistent, with outbreaks often occurring unpredictably. The ongoing battle between viruses and host immune systems is a dynamic and ongoing process of evolution. Viruses have evolved countless strategies to evade immune responses, including mutation, latency, and even altering the host's immune cells. One example of this is the Epstein-Barr virus, EBV, which is linked to mononucleosis and several types of cancer. EBV has the ability to hide in the host's immune system by targeting and infecting B cells, ensuring that the virus can persist for life. In some cases, it can even cause the body to mistakenly attack itself, contributing to autoimmune diseases. Some viruses can even affect the way a host organism develops. The Zika virus gained notoriety for its connection to birth defects, particularly microcephaly in babies born to infected mothers. The virus affects fetal brain development during pregnancy, leading to severe neurological disorders. This devastating effect underscores the power viruses can have over not just health, but the very course of life. Zika's influence on brain development has led to a greater understanding of how viral infections can interfere with normal biological processes during crucial stages of development. On a more microscopic scale, viruses often function as the ultimate evolutionary drivers. They can transfer genes across different organisms and species, facilitating the spread of new traits through populations of bacteria, plants, or animals. This gene transfer can occur both within and between species, influencing traits such as antibiotic resistance, viral immunity, and even behaviors like aggression. The role of viruses in shaping genetic diversity is often underestimated, but it is clear that they play an essential part in the evolutionary processes that define life on Earth. One of the most intriguing aspects of viral biology is the way in which they are categorized. Viruses are classified based on several characteristics, such as their shape, type of genetic material, DNA or RNA, and how they replicate within host cells. The Baltimore classification system is one of the most widely used methods for categorizing viruses, dividing them into 7 distinct classes based on the nature of their genome and replication strategy. This system not only aids in understanding viral evolution, but also helps in developing antiviral treatments that target specific viral replication mechanisms. Viruses can often play an unexpected role in agricultural research, particularly in the development of genetically modified crops. Agrobacterium, a bacterium often used in genetic engineering, is a type of microorganism that transfers DNA into plants. In some cases, the gene transfer process closely resembles viral infection. Scientists have harnessed the ability of viruses to transfer genetic material, allowing them to insert desired traits into crops. These viral-like processes have enabled the development of genetically modified plants that are more resistant to pests, diseases, and environmental stresses, contributing to food security in various parts of the world. In the natural world, viruses can have a profound impact on plant ecosystems. The tomato mosaic virus, TUMV, for example, infects a wide range of plants, causing a mosaic pattern on the leaves and stunted growth. While this virus does not generally kill plants outright, it can severely reduce crop yields, causing economic losses in agriculture. Infected plants often exhibit chlorotic lesions and a decrease in photosynthetic activity, making them less productive. Such plant viruses highlight the subtle and often underappreciated ways in which viruses can shape the environment and agricultural productivity. Viruses can evolve very rapidly, which is one of the reasons they are so difficult to control. The high mutation rates of RNA viruses, such as HIV and SARS-CoV-2, allow them to quickly adapt to new environments and evade host immune defenses. This rapid evolution is also what makes it so challenging to develop vaccines and antiviral drugs that remain effective over time. Each new generation of virus is slightly different, meaning that the immune system may struggle to recognize and fight newer strains, necessitating the constant updating of vaccines and treatments to keep up with evolving viral threats. While viruses are typically seen as harmful invaders, they are sometimes involved in processes that benefit their hosts. For example, some bacteriophages help regulate bacterial populations in environments like the human gut, ensuring that no single species of bacteria dominates. By selectively targeting and killing harmful or overly abundant bacterial strains, bacteriophages contribute to maintaining a healthy microbial balance. This process is essential for the overall health of the microbiome, which plays a crucial role in digestion, immune function, and disease prevention. One of the ways that viruses manage to evade the immune system is by altering their structure. The influenza virus, for instance, frequently changes the proteins on its surface through antigenic drift and shift, allowing it to escape immune detection. This variability in surface proteins is why people can get the flu multiple times, and why new flu vaccines need to be developed each year. The ongoing mutation and adaptation of viruses like influenza are a constant challenge for public health, highlighting the need for flexible and responsive vaccination strategies. The idea of viruses as biological weapons has been explored for decades, with some viruses being weaponized for use in biological warfare. The potential to harness viruses as tools of destruction raises ethical and practical concerns, as viruses can cause widespread devastation, especially if they are intentionally released into a population. Viruses like smallpox, anthrax, and even botulism toxin have been studied for their potential use as bioweapons, although international treaties have since banned the development and use of such biological agents. Despite the ban, the possibility of viral bioterrorism remains an area of concern. In the world of research, viruses have proven to be invaluable tools for studying the inner workings of cells and molecular biology. Adenoviruses are frequently used in gene therapy to deliver therapeutic genes to target cells. These viruses have a unique ability to infect a wide variety of cell types, making them useful in the treatment of genetic disorders. Similarly, viruses like retroviruses have been crucial in the development of recombinant DNA technology, a cornerstone of modern biotechnology. By studying how viruses interact with their hosts, scientists can develop new ways to treat genetic diseases and even cancer. Viruses are constantly in the process of evolving and adapting to their environment, which is why they often exhibit extreme levels of genetic diversity. One example of this is the hepatitis C virus, HCV, which has such a high mutation rate that it can develop new strains resistant to the immune system or antiviral treatments. HCV's genetic diversity makes it difficult to create long-lasting vaccines or treatments. The constant evolution of viruses like HCV is one of the reasons why chronic viral infections remain a significant challenge in global health despite advances in medicine. The genetic material of viruses can be incredibly varied, with some viruses containing DNA and others containing RNA. While DNA viruses replicate their genomes using the host's cellular machinery, RNA viruses have to reverse transcribe their RNA into DNA before replication can occur. This unique process is used by viruses like HIV to integrate their genetic material into the host cell's genome. RNA viruses tend to have higher mutation rates than DNA viruses, which contributes to their ability to evolve quickly and evade immune defenses. This rapid mutation is also why RNA viruses, like the flu and the common cold, are so difficult to vaccinate against. Many viruses rely on vectors, such as mosquitoes or ticks, to spread from one host to another. The Zika virus, which was spread by Aedes mosquitoes, is one example of a vector-borne virus that caused widespread illness. These viruses typically affect both the insect vectors and their mammalian hosts, creating a complex web of interactions that makes controlling the spread of the virus difficult. As climate change continues to impact global weather patterns, the range of many mosquito-borne viruses like Zika, dengue, and malaria is expanding, potentially putting millions more people at risk. The complexity of viral evolution is seen in the phenomenon of viral recombination, where different strains of a virus mix genetic material to create entirely new virus variants. For example, during an influenza outbreak, two different strains of the flu virus can infect the same host cell and exchange genetic material. This can lead to the creation of a hybrid strain, potentially more dangerous or more contagious than its predecessors. The ability of viruses to recombine makes them highly adaptable, ensuring their survival across generations. This process is one of the reasons why the flu vaccine must be updated annually. Viruses are sometimes employed in ecological studies to track animal populations, particularly in studies involving wildlife conservation. For instance, researchers have used viruses like the canine parvovirus to trace the movements of wild canid populations. When animals are infected, they carry viral DNA with them, which can be identified in their droppings or saliva. This allows scientists to monitor populations of species like wolves, foxes, and coyotes over vast regions without needing to trap or directly interact with the animals. It's an innovative use of viruses in non-disease-related research. Not all viruses infect living organisms. Some viruses, known as viruses of the dead, infect decomposing organic material. These viruses infect bacteria that thrive in decaying matter, contributing to the breakdown of organic material into nutrients. This form of viral infection plays a role in the decomposition process and is essential to nutrient cycling in ecosystems. Without these dead viruses, the breakdown of organic material would be much slower, impacting soil health and the availability of nutrients for plants and other organisms. Pox viruses, a family of large complex viruses, have had an outsized impact on human history, especially through their role in the eradication of smallpox. The smallpox virus was once one of the deadliest viruses known to humans, causing widespread epidemics throughout history. In 1980, the World Health Organization declared smallpox eradicated thanks to an intensive global vaccination campaign. This achievement stands as one of the greatest accomplishments in global public health, and it is a testament to the power of vaccination in preventing viral disease. The human immunodeficiency virus, HIV, which causes AIDS, is unique in that it specifically targets the immune system, weakening the body's ability to fight off infections and diseases. What sets HIV apart from many other viruses is its long-term persistence in the body. HIV infects CD4 plus T cells, a type of white blood cell that plays a key role in the immune response. Over time, the virus gradually destroys these cells, leaving the immune system compromised. This progressive damage makes HIV one of the most challenging viral infections to manage, despite significant advances in treatment. Interestingly, not all viral infections result in symptoms. Some viruses can infect hosts and remain entirely asymptomatic. For example, the Epstein-Barr virus, EBV, responsible for causing infectious mononucleosis, often remains dormant in the body for long periods after the initial infection. Most people infected with EBV never develop symptoms, though the virus can occasionally be reactivated, causing illness later in life. This asymptomatic persistence is a hallmark of certain viruses, and it can complicate efforts to control and eradicate viral diseases. The concept of reverse zoonosis is a growing area of research where viruses are transmitted from humans to animals. While the reverse zoonosis concept may sound unusual, it's increasingly common in the context of emerging viruses. For example, SARS-CoV-2, the virus responsible for COVID-19, has been found to infect various animal species, including minks and tigers in zoos. This phenomenon suggests that viruses can jump from humans to other animals, which could lead to the development of new variants or further spread among different species, complicating efforts to contain outbreaks. Viruses are among the most ancient forms of life on Earth, with some researchers suggesting they may have existed for billions of years. Their ability to evolve rapidly, mutate frequently, and adapt to changing environments suggests that viruses may have played a crucial role in shaping the course of evolution itself. By introducing genetic material into host organisms, viruses have influenced the genetic diversity of both prokaryotic and eukaryotic life forms. Some even believe that viruses may have been instrumental in this emergence of complex life, possibly acting as intermediaries in the transfer of genetic information between different organisms. Some of the world's deadliest viruses, such as Ebola and Marburg, belong to a group known as filoviruses. These viruses cause hemorrhagic fevers characterized by widespread bleeding, organ failure, and high mortality rates. While rare, outbreaks of these viruses can be devastating, particularly in regions with limited healthcare infrastructure. Filoviruses are often spread through direct contact with infected bodily fluids, which makes containment efforts particularly challenging. The ongoing threat of these viruses has driven much research into better diagnostics, vaccines, and treatments. The use of bacteriophages in modern medicine is showing increasing promise as an alternative to antibiotics. With antibiotic resistance on the rise, researchers are revisiting bacteriophages as a solution to treat bacterial infections that no longer respond to conventional antibiotics. Bacteriophages are highly specific, targeting only the bacteria that cause the infection without harming healthy bacteria in the body. This specificity makes phage therapy an attractive option for treating conditions like chronic sinus infections or wound infections, especially in cases where antibiotic treatments have failed. As our journey through the hidden world of viruses comes to a close, allow yourself to sink even deeper into relaxation. Let the gentle sensations of peace and calmness envelop your body and mind. The mysteries of life in all their complexity are part of the vast and beautiful tapestry that surrounds us. Feel your thoughts gently fading, your muscles softening, and your breath flowing naturally and effortlessly. Rest here, fully supported in this safe and serene space. If you enjoyed this relaxing moment, please consider leaving a like, subscribing, or sharing your thoughts below. Your support is deeply appreciated and helps others find their way to this peaceful space. You can also turn on the notification so you don't miss any of our future journeys together. But for now, let your body relax, release any tension, and allow your mind to drift. Good night and sleep well.